So this talk is probably not going to be as philosophically and even as biologically concise as I would have wanted, but I still hope it's going to give you some interesting intuitions on how to think about development. So the title of the talk is Normativity and Plasticity in Development, and specifically I'll be focusing on some perspective from Michael Levin's work on morphogenesis. So this is a frog embryo, programming itself you can see cells moving and constructing a form, right? And you have lots of these examples in development, and of course, this process starts from a fertilized egg. So basically, one cell, and then you get to all of these complex dynamics. And so this is just some quotes from the developmental textbook. Multicellular organisms do not spring forth fully formed, rather they arise by a relatively slow process of progressive change that we call development. And this other one is kind of fun. Every multicellular organism has to function even as it builds itself. It develops. As an embryo, you had to build yourself from a single cell. You had to respire before you had lungs, digest before you had a gut, build bones when you were pulpy, and the last one is probably the most problematic for me, form orderly arrays of neurons before you knew how to think. So this is kind of the overview of my talk. First, I'm going to talk briefly about the philosophical framework I'm coming from. Then I will shortly talk about the context of Michael Levin's work and who is he. Next, we will talk about where is the organ evolution and development, specifically in development in this case, but it seems that even in biology of the 20th century, the organism was kind of put aside. Then I will talk about Levin's idea of navigation of a problem space as a sort of invariant in different levels of biology and cognition, and levels of competency, starting with single cell competencies, and then multicellular competency or competencies of cell collectives, defining morphogenesis as navigation through a morphospace, and then also about how the process is plastic, the set point or the goal it tries to reach is plastic, and then very briefly, you will have time, the ontological status of these anatomical set points. There's a lot of slides, there's a lot of pictures, so we'll see. Okay, so this is just briefly about my framework that I'm thinking in. So the top right to top left philosopher is Wilfrid Sellars, and he kind of describes these two conceptual images of the world. One is a scientific image of the world, and one is the manifest image of the world, and this is very broadly and idealistically conceived. And I'm not going to really talk about is the scientific, ontologically primary or the manifest, or which is more real. For now, I'm gonna have a more neutral approach to this. But the goal of Sellars, or he thought that the goal of philosophy was to articulate one synoptic vision, where you can conceive of phenomena as through the scientific image, but also you have to reconcile it with certain aspects of the manifest image. And for him, the aspect you need to account for, even in the to reconcile, is the normative aspect. So Voros before said that even Sellars talks about Janus-faced phenomena, and this is normative phenomena are for him Janus-faced in the sense that you can at least describe them or conceptually think about them in the causal realm, but also in a normative realm. So using things like ought to and should and stuff like that. And the other two are, of course, Maturana and Varela, and this will be poignant for the last step of this presentation. So the status of these set points or this whole process, and I will try to link this with modern developmental biology, that is kind of empirical biology of Michael Levin, and they are explicit in saying that the understanding of the organism is possible if we highlight the autonomy of the living. Okay, so this is Michael Levin. He's a developmental and synthetic biologist at Tufts University. You probably have seen him recently because he's become really like a popular public speaker. His like areas of expertise are developmental biology, and he kind of combines it with cognitive science and computer science. But like from the cognitive science view, he's not like, I would say, like he delves into more these traditional approaches in cognitive science, so cognitivism, computationalism, information processing, using techniques from neuroscience, and like really likes cybernetics as well. And he has a distinctly engineering perspective, which I think is good while he sticks to it, but sometimes, like especially in the last talks, he's trying to present a more unified approach, and then you get into this murky philosophical claims. But okay, let's just stick with that he has this engineering perspective and that it can tell us something about living systems. So the question, where is the organism? On one hand, like we had in the 20th century, like a rise in molecular biology, so biology that focuses on like molecular mechanisms. And on the other, we have a lot of like biology that focuses on the population and like evolution taking place on the level of population variation. And so, where is the organism in this picture? Or is an organism just an array of physical and chemical processes that are like acted upon by natural selection? Or does it play perhaps a more active role? And Levin said that there are two ways of bringing back the agent. One is through like organism level learning and intelligent behavior. This is the more common thing. And what he does is like talk about more suborganismal physiological and anatomical level intelligence. And also like he proposes then a sort of unified framework to how to maybe think of all of these levels of competency in one framework. Okay, so basically we all like start like from this like, or not, most organisms start from like a fertilized egg, right? And then you get this very complex structure. You go from like a genotype to the phenotype, and typically like in traditional modern synthesis you said, okay, genotype is like a site of mutation, and then you go to phenotype, that which selection acts upon, that which selection sees actually. Of course, there are several waves of biology that like undermine like only these two processes, epigenetics, whatnot, but we're not going to talk about those specific things here. But basically, we're again at this problem of like that Gregor talked about, or like several others, that you have like these genes, and then you have like proteins that are like the active molecules in the cell, and then some sort of physics happen, and then some sort of emergence happens, and then you get an organism. And like, can the behavior and development be viewed just as this exclusive dynamical system perspective in which different outcomes simply occur as the result of different environmental inputs working through a mechanical process? So this very linear conception of just a mechanical process unfolding. This is like how developmental biology usually like try to describe these processes. So like this is what I learned at faculty. You have like gradients of gene expression. This is a fruit fly embryo, and like you have a gradient of one gene and a gradient of the second gene, and then you have because of this you get some sort of like a strip. You can like differentiate the anterior and posterior, so where the head will be and where the tail would be, basically. So, but you start with like, okay, you have like just these gradients, and then they determine more and more complex, and it's like this information processing on the level of patterns of gene expression. And Levin points out that this is actually an indirect relationship, right? Genomes do not code for anatomies directly. There is like a control layer between a genome and a phenotype. And this control layer for him is like morphogenesis. So morphogenesis is the creation of ordered form. And his idea is that competent navigation in arbitrary problem spaces is an invariant for thinking about scaling of cognition during evolution. So you have different problem spaces, like here you have like navigating a 3D space is basically behavior for him. And you can think about this sort of problem spaces on different levels. So you have like a physiological, a metabolic, a transcriptional, a behavioral problem space, and you have to like traverse that space. And for instance, in morphogenesis, you would have a morphogenic problem space or morphospace, which are like all possible morphological states of a genome, for instance. But we can like discuss if this is it has more implications. So this is a planaria which has a very regenerative potential for a head. So you cut off their head and they grow another one, like all the time. But you can also like by like just 3 genetic factors, you can like kind of direct in which way their head will form. So you know you can get this is the traditional planaria, and then you can get all of these weird shapes. So how do cells navigate this morphospace? So Levin says, okay, cells are not passive. Are not passive particles, right? Cells have a lot of competency. This is a single cell organism that's like here is like trying to eat this other thing, but it can like move, it can hunt, it can proliferate, and as we saw in the beginning of the frog embryo, like in the process of morphogenesis, there's a lot of like moving and inverting and trying to form structures, right? So cells in themselves have a rich behavioral repertoire. Okay. And yeah, this is just some quotes from Levin. Cells in their competence have many competencies, like the ability of gene regulatory networks and chemical pathways to learn from experience, rapidly adjust to compensate for entirely novel physiological stressors, and they even have like unicellular slime molds that are also kind of famous now, have this capacity to explore environment at a distance and make decisions using memory and comparison of alternatives. And so basal cellular competencies did not disappear just by them going into a multicellular form. Instead, they scaled up as a collective intelligence to operate in larger and more complex problem spaces. And this promote like, so this sort of informational integration helps to wipe, like this is an interesting statement, but helps to wipe the individuality of cells and enlarges the memory, predictive capacity, and sensory effector radius of cellular networks, enabling them to pursue larger goal states in spaces like anatomical morphospace. So how do cells then know what to do and where to stop? So how does, just from being in a certain position in an embryo lead to, I don't know, formation of a limb, formation of an eye, formation of all these things? And so, yeah, this is just a picture from this quote. So you have like a single cell, and then you get them integrated in some way, and this produces like an expanded like goal representation. And we're going to talk a bit more about this now. Okay, so Levin's idea is intelligence of cell collective to reach an anatomical homeostatic set point in spite of perturbations. And this is like the same picture as before, but now you have, of course, a loop, like the favorite thing of these dogs. So a closed loop pattern homeostasis. So basically, the point is that you have some sort of representation of a future state, of a blueprint of where you want to move to, and the cellular collective uses this, or like it can compare its current status to this like representation and adjust its navigation towards that future state, like by way of like some sort of comparison. So cells and tissues change position and shape, gene expression, until the correct shape is reestablished, and then they stop. So how is this correct shape like embodied in the organism? Like where is it? And Levin's idea is that you have like homeostatic points you have anatomical homeostatic set points that are, at least on some occasions, or like that's an example of their embodiment is in bioelectrical networks. So not only brains and neurons have like bioelectrical networks, like all the cells of your body can produce like a potential energy, right? And so they can group together using these bioelectrical networks in just like a information glue in a way. And then he uses different like interchangeably. So anatomical set point and bioelectrical network are really like how we think about in this scientific image, for instance. But then he talks about this being like a pattern memory to which you like go, or a representation of a future state, or a representation of a goal. And so cells navigate towards that future state in a plastic way. So yeah, I'm going to talk about two levels of plasticity here. One is the plasticity of the process itself. So this is this like enabled by this loop. So this is basically that you reach the same representation of the future goal, even though you have like perturbations, like injury, or just like perturbations that happen in not just in scientific context, but also in nature. And then I'm going to talk about how Levin's work also shows that the goal, like the representation of the goal itself, is plastic. It's not as fixed as like usually when we look at development, you think, okay, you go from cells and then you have a very like robust process of going to the organism. But Levin shows that this is only like, it seems like there is only one pathway, but there are actually many pathways and many set points that cells can reach. Okay, so the plasticity of process is to reach the same goal in spite of perturbations to the system. It entails some kind of downward causation in the service of this large-scale anatomical goal. Different molecular mechanisms, like cell-cell communication, cytoskeletal bending, and whatnot, are deployed as needed for an unexpected change. And this is also very important for a conception of like evolution, because this ability gives organisms it gives them the ability to handle an unexpected alteration without period of evolutionary change. So again, it's not like you're just a passive organism and then you're waiting for your random mutations to occur and then maybe you die or maybe you don't. But the idea of Levin is no, actually evolution evolves problem-solving agents, right? So it's not a passive material. It can readily adapt, and also in a way that means that there's a interconnection with evolution, because we can talk about that later. Okay, so the plasticity of process. So yeah, usually you go from like this in a very regulative manner to like a very complex, right, developmental. This is like a section of some tissue with a lot of form structures. And yeah, usually we think about it like, okay, so you go from one state to the goal, right? But actually, you can start at very different starting positions and get to the same goal. So one example is, this is what they did in the lab. They take a tadpole and they like did a Picasso-like tadpole. So they took its cells and they like just randomly rearranged them of the face, and you can get both of these like starting positions end up in a normal frog like face. So the cells still know how to like reach the same goal that is still like represented in the. Then another like example is if you like change this bioelectrical network like randomly on an embryo, you can induce eyes or like different structures from cells that were not like, that were, if you would leave them, they would turn out into, I don't know, gut. So you can induce structures by like saying, okay, this is the set point of an eye, and you say to cells, you just give them the set point, and the cells themselves don't like micromanage, but like they know just by having that electrical network what to build, and that's an eye. And even more interesting is perhaps this, that even if you like, so you modify only like a bunch of this bioelectrical network, but these cells then also include some others because they realize that it won't be enough for just them to build an eye. So they realize, okay, I actually need more if I want to reach this set point that is the eye. So I'm going to like recruit just cells that were actually not modified by this network. So this is just, the process is more than just triggers of master regulator signals. When an insufficient number of cells is modulated, they recruit their normal unmodified neighbors to complete the task of building an ectopic lens of the right shape and size. This ability to complete a particular action in morphogenetic space by controlling the behavior of other cells in a context-sensitive, dynamical manner is a key feature of controllability and reliability. It's how the problem-solving capacity of cells, not just ability to execute the same hardware or local rules, facilitate adaptive robustness in novel circumstances. Okay. This is another form of a plastic process. So the axolotl that already Jan mentioned yesterday is a very popular model organism, and it's most known for its impressive regenerative capabilities. So basically, if an axolotl loses its arm in nature or in the lab, it's able to regrow a functional limb. So basically, again, this is like Levin shows that this is because they have like this pattern of the bioelectrical signals, and then they just like the cells work again towards this future representation. And again, this is just maybe interesting. Again, okay, so this is like the development of an frog embryo, and these are like the bioelectrical signals that like then guide, I guess, these things. But this maybe is more of an open question. I just put it here because it's interesting that Levin says that we can think of like developmental progression as a collection of regenerative repairs. Each stage is in effect a birth defect from the perspective of the subsequent stage, and is repaired by regulative development, which seeks to minimize error. And this perspective hypothesizes that the bioelectrical target pattern changes more rapidly than the transcriptomial and anatomical patterns, and pulls them along by stress minimizing loops. And again, the open question for me here is like, okay, then you go into like maturation and adulthood, and this biochemical and bioelectric pre-patterns stop changing. So why do they stop changing in adulthood? And probably you can like again invoke some sort of representation of when to stop, not just the development of limb, but like when are you an adult form, right? And another interesting thing is, okay, so this is again these cells that are like connected through an informational network. But we can talk about the difference also between like a collection of cells versus a cell collective. So what's the difference between just cells together and like a proper cell collective? And we can see that if you, for instance, like cut off one cell from this bioelectric network, it induces cancer. So basically, what it means, or how Levin interprets this, is that, okay, these cells are like aligned to reach the same goal. They sort of have the same purpose. They are constrained by the same goal. But once you are cut off from this network, you move back into this individual single cell goal direction. And that's just like cancer cells are like notorious for like becoming very undifferentiated and then just like replicating rapidly, right? And yeah, so Levin talks about alignment of purpose, and the cells align their purpose to a common goal. So they are constrained by the pattern, and so cancer would be back to single-cell purposes. And I'm gonna explain a bit why I'm talking about purposes here, because it has to do with an action, I guess. Okay, so now just briefly about also the plasticity of the goal itself, which is basically means to change this set point by modulating the bioelectrical network. So first few examples are just by scientific modulation of this, so human intervention. So frogs are not axolotls. They cannot in nature repair their legs. If they lose a leg, they lose a leg, and that's it. But what Levin found, or Levin in his laboratory, that you can induce regeneration of the limb by doing this, it's cocktail treatment with basically what you do is induce bioelectrical networks through ion channels. So by inducing this certain bioelectrical network, so a certain representation of a set point in a frog, you can produce something that you do not find in nature. You kind of trigger that the frog cells, which are competent, then reach or like move towards the goal of the limb, and you get not only like just tissue regeneration, but it's a functional limb, right? So basically, you take the set point and like produce the same result in a different species. Another thing is, so this planaria, when we talked about before, that if you like cut them in half, they grow like, if you cut their head off, they grow another head. Like they're basically immortal like that. And Levin also showed that you can do like, if you like cut off the head of a planaria and then do some bioelectrical perturbation here, so you kind of change the set point, the bioelectrical network, you can get basically different shapes, and these shapes are very like similar to other species of planaria. So basically you get different, just by tweaking this, you get different shapes of different species of planaria that are like millions of years like separated in evolution. And another thing is like the plasticity of the goal. It's not only done by humans, but not only by human interventions, but also other organisms can also like use these mechanisms and target them to like produce different forms in other organisms. So this is a leaf that usually doesn't have this, but a certain wasp injected something in it and like changed the bioelectrical network, and by doing so, the leaf itself produced these very wild things, and later they will fall to the ground, and they have little, like the wasps put their little eggs inside, and the wasp reproduces. So the point is that other organisms are targeting, not like micromanaging like some genomic or like molecular states, but they're targeting these high-level regulatory mechanisms. And okay, then this idea from Levin is that, can we talk about a unified competency here at different scales? So one is like transversing like metabolic space, and then physiological, transcriptomal, amorphous phase behavior, and then we can go even more up. But also the idea is that the evolution kind of pivots these different ability to transverse these different problem spaces. Okay, so now I think we have time that I can finish this. This is going to be the more speculative part. So Levin talks about goal-directed behavior, but does he really mean goal here in this like philosophical way sense, or is it just like anatomical set point directed behavior, right? So it's just a constraint and you're just like, okay, moving towards it. So what is the status of these anatomical set points? And here I'm invoking again this distinction that Zellers talks about of normative phenomena, where you can have the same process described as a bioelectrical network, so in the realm of causes, but you can also describe them as a representation of a desired form or a future goal or a norm you're moving towards or you're measuring your current state towards. And so you can also talk about these processes like in the realm of norms or wholes, right? So then I am like referencing a bit the paper by Weber and Varela, so Life After Kant, in which they try to propose to bring back the notion of teleology into speaking about biological systems. So they do not agree that we can, like, this is just that we are speaking of goal-directed behavior only as a heuristic until we find a better explanation. So it's going to be a mechanical process, maybe this is it, but we should stop like invoking any kind of goal-directed behavior. And then Levin uses a lot this concept of interface, or affordances even, for basically defining this set point through interactions other organisms can have to it, and evolution can have to it, and also the organism itself has to it. And then I will talk a bit about vital norms quickly. So Levin says that, for me it's really interesting because he says for the same reason it limits, so the same reason why bioengineers are now like trying to perturb these processes, not through micromanaging like molecular states or genomic states, but through like these higher level states. The same reason is that that evolution targets this like level, not like lower levels. So it has like, in a way, its existence is there because it's ontologically maybe real in this sense that other processes use it to exert their effect on it. And again, okay, so he talks about a control interface where you have some low complexity input and you get a very high complexity output. And these are like affordances for engineers and evolution and other organisms. So you still have this, you know, it's still just mechanics in some way, right? It's still a physical process. It's still a causal process, but this new domain is acted upon, and it can be acted upon by scientists, by evolution, and by other organisms, and of course by the organism as a cell collective itself. But Levin here just like presupposes, okay, I think in one of his slides he actually says, okay, we go from just physics to emergence to like cell is just physics, and then you go to like higher levels. But so the point of enactivism that cells themselves are not just physics in this mechanical sense. We can talk about an activistic like natural purposes. So this is like an intrinsic teleology that arises from biological autonomy and biological individuality. And Jonas uses the phrase, the dialectical priority of form over matter is an ontological surprise. So it has an ontological status that arises from this formation of identity. So this is, maybe we can skip this. But also I went and checked in Tree of Knowledge, right? This is one of the earliest books of Maturana and Varela. This is the representation of an autopathic system. And then they talk about metacellularity, where you basically get, from this you have this connection, and this can go either to like a symbiotic relationship, which is how it's hypothesized that basically mitochondrial, like eukaryotic cells were evolved. But you can also go to this other direction, which is like a multicellular existence, like a collective of cells. And in this book they says, okay, this is like published in, I don't know, in the... Okay. So they say that for the purpose of this book, we shall leave open the question of whether or not these metacellular systems are first-order autopathic systems. But what we can say is that they have operational closure in their organization. Their identity is specified by a network of dynamic processes whose effects do not leave that network. And whatever the organization of these metacellulars may be, they are made up of first-order autopoietic systems, so cells, and form lineages by reproducing through cells. So these two conditions are sufficient to assure that whatever happens in them as autonomous unities happens with conservation of the autopoiesis of their component cells, as also with the conservation of their own organization. So their organization, even for Maturana and Vela, consists of an identity. I think this is probably the last slide. Just this idea why is cybernetics enough to explain this. So Jonas says, okay, no, we have to include concern into, we have to invoke the notion of concern if we want to really explain life. We can't just say, okay, this is a very complex homoaesthetic process. And I think this is a similar notion here in this article, Life After Kant, where the point is that, yeah, form then is not just an abstract goal in a genetic program, but a material task to fulfill from moment to moment. The genetic program influences form, but only in being interpreted by the soma according to the actual needs of self-maintenance. And okay, actually, this is the last one. Yeah, this is actually the proper slide. So it's like autopoiesis is just one more self-organizing mechanism, which is undoubtedly is. But Varela and Maturana say other self-organizing mechanisms stay within the perimeter of a physicalist view of nature. So they stay in this only describing this causal realm, as Sellers would say. But there is an essential difference between autopoiesis as a self-organizing concept and other self-organizing concepts, which is this radical transition to the existence of an individual, a relation of organism with itself, and with this, the origin of concern based on its ongoing self-produced identity. So this kind of has to do with what I was asking before, like where does this point of interiority come into the picture? And yeah, I think it nicely ties up with the presentation before mine. So that's it. Thank you.